Okay, so it's 9 a.m. We'll go ahead and get started. Good morning, everyone. I'm Justina Miskovich. I'm the honorary editor of the Proceedings of the Royal Society of Queensland in 2022. I'll begin by acknowledging the traditional custodians from whose lands we're tuning in for this event today. And I will hand over to Bridget Chili, who has very kindly agreed to do our welcome to country.
Thank you. Thank you. Wanya Nalam, everyone. My name is Bridget Chili. I identify as a Marula Kabi woman on Judingar country here on the Sunshine Coast. Yeah, I'm on matriarchal country, and I just wanted to say thank you to the Royal Society for actually having this opportunity and to be able to, do, this action of reconciliation, to be able to do welcomes to country. So I'm very grateful for that. So on behalf of my ancestors and my old people, I'd just like to welcome you to this beautiful meeting today. Thanks.
Thank you so much, and thank you so much for joining us. I will now hand over briefly to Dr. Ross Hines, who is the president of the Royal Society of Queensland, to do a short introduction.
Thank you, Justina. It gives me great pleasure on behalf of the Royal Society of Queensland to extend a most warm welcome to all participants in this important showcase of early career researchers projects. I offer special thanks to you, Dr. Justina Miskovich, for your excellent leadership and organisational skills in convening this exciting program. Also, special thanks and encouragement are extended to all presenters and, where appropriate, their collaborating researchers. The scope and diversity of the papers is most impressive and motivating. I wish you every success in your presentations and longer-term research outcomes. Best wishes to all for a lively and stimulating showcase program. Thank you.
Fantastic. Thank you so much. So our first talk will start at 10 minutes past 9. So I just wanted to very briefly go over through some of the online housekeeping. Most of you would have already seen our brief that got circulated around to registrants and to speakers, but just want to make sure we're all on the same page. So just a reminder, friendly reminder to everyone, please stay muted and have your camera switched off unless you're presenting or asking questions. And this is just to limit background noise and to make sure that our bandwidth, is kind of okay, because we've got obviously tens of people joining us today. Now, feel free to use the chat function that you see in your Zoom settings throughout the event to post comments or your questions. And I guess what this means is you don't necessarily have to leave your questions to the end of the kind of Q&A part of the speakers' talks. And in the audience today, we have members of the public, scientists from across Australia, and of course, members of the Royal Society of Queensland Council. So we've got a range of people joining us, different backgrounds, and hopefully we can kind of facilitate a very lively and exciting discussion and engagement, as Dr. Hines mentioned earlier. I should also clarify, in the brief that we sent around, I initially mentioned that you should be able to click on the raise your hand icon in your Zoom panel if you wanted to ask a question, but we realized that we're actually in a Zoom meeting, so there isn't that option. So again, just feel free to pop your questions or comments in the chat function, or simply you can speak if you want to. The hand icon is typically for the webinar settings. What else? So we'll be recording the presentations today, unless any of the speakers have specifically asked me not to do so. And I would also ask that you please respect that if a speaker says, please do not share any of the results or the slides, on social media and so on, because some speakers apparently have results that haven't yet been published or peer reviewed. So we're sort of just using this as an opportunity to chat about the work. And I will also clarify that the Q&A sessions after the talks will not be recorded. So those sessions and talks that will be recorded, I'll later upload them onto YouTube. They will be accessible publicly, but the Q&A discussion at the end won't be seen in that recording. I will run the first session, which starts in just about 5 minutes. And then the second session, after a short break where I encourage everyone to grab a cup of coffee or water, you know, do whatever you've got to do to take a break. The second session will be run by Dr. Peter Dart, who is also in the audience. And I guess one other thing I just wanted to mention was to all presenters, Pamela Lauder, who is also in the audience. Do you want to give us a quick wave, Pam, so that everyone can see you? This is Pam. We'll give you a short warning at 6.5 minutes of your allocated 7 minutes for your presentation, alerting you that your time is almost up. And so please try and not, you know, don't stress too much. It's just sort of letting you know the time is almost up and you're possibly going into your Q&A time. So obviously we want to try and avoid that. And so that's really about it from me. We've got about 5 minutes until the first talk, so we'll let other registrants kind of roll in. I will, just minimize my screen for now, and then in about 4 minutes, we'll have Eva Martellotta from Griffith University giving her first presentation. So I'll see you all in a couple of minutes.
Yes, I'm just going to make a comment before we get into the session that I'm involved in. I'm rereading the abstracts and they are so varied, it's wonderful. And I'm learning stuff as I go. I felt I had a pretty good general knowledge about things biological and scientific, but we're going to have some very exciting stuff actually.
Yeah.
Some of it I'm aware of. I'm just looking at Alexandria's abstract, for example, and that's something that I've been following and involved a little bit in. But the rest of them, thinking, wow, this is really interesting stuff. Particularly, one of them, another one just struck me because I read it just now, is the modeling of coral reproduction. We've got lots of interesting stuff to talk about this morning.
Absolutely. I think that's definitely the highlight for me as well, just a range of topics. Great.
Yes,
like I'm going to learn so much.
Absolutely mind-blowing.
Yep, absolutely.
And for all of those involved, including non-speakers, please let your colleagues know that this is a great opportunity to have a practice at giving a talk. Yeah. And that's part of your scientific training nowadays, particularly. And that if we get such a good feedback from this morning, we should also think about running it again. And Justina and I have just been talking about running a 3-minute session symposium. So all those things are up for grabs, depending upon how people think about this morning. I'm really excited, as you can hear.
Absolutely. I am really excited as well. And it's such a shame we can't do it in person, but I'm really glad that, you know, we're able to do it online anyway and engage and interact and make contacts. Definitely. Eva, would you like to perhaps already share your slide and then we'll get started at 10 past?
Yes, sure.
See if everything's working. Excellent. Thank you.
Yeah. Should I share my screen or do you have to authorize me? Okay.
Yeah, I think you should be all good.
I've
changed the settings so that everyone should be able to share.
Yes.
I think we can see your screen now.
Yes.
Excellent. All right, so let's just wait 1 more minute and I'll just introduce you and then we'll go ahead and get you started.
Thank you.
Yeah, I'm going to wrap it on again. the other thing that really got me going was the fact that we've got such varied institutions involved and research groups, and it's so broad in its scope. It's another wonderful thing, and wonderful thing for the Royal Society that, of course, publishes the proceedings that are an opportunity for people to publish, and it's a very prestigious journal. It's the oldest in Queensland. So
okay, Peter, apologies. I'm going to stop you there so we can stick to schedule, okay? So it's 10 minutes now.
Good.
All right, thanks, Peter. Okay, so without further ado, everybody, it's a real pleasure to be introducing our first speaker of session 1, Eva Martellotta, hopefully I'm pronouncing that correctly, apologies, from Griffith University, who will talk to us about boomerangs, shaped stone tools in ancient Queensland, experimental archaeology and traditional knowledge reveal a new function of the most iconic Indigenous tool. Go ahead. Thank you so much.
Thank you. Hi, everybody. I'm Eva Martellotta, and I'm just going to talk about my PhD research topic, which is the use of boomerangs in ancient Queensland and ancient Australia to shape stone tools. So the first thing I want to talk about is the question why we shape stone tools. It's very easy. It's just to change their functionality, which sounds like a fancy concept, but actually you can think about something that we have, in our everyday life. The difference between a chef knife and a bread knife. As we know, we use these two types of knife to cut different materials. That's because the chef knife has a straight blade, so it's more functional for specific type of food, and on the other end, a bread knife has a serrated blade, and it's more functional to cut other types of food. And this is a concept that goes way back, because we have heaps of stone tools in the past, which show actually the same concept. So we have stone tools with a straight blade, which were used for a certain type of function. And then we have other stone tools with a different edge, a modified edge, which can be associated to the serrated edge of the bread knife, for example, which was specifically shaped and modified in order that so the tool can perform different functions. And this action of modifying the edge of the stone tools, in archaeology, we call it retouching. So in Europe, where I come from, we know a lot about retouched lithic tools. We know that, first of all, we have so many of them in so many different archaeological sites. We know that they were retouched using some specific pieces of bones, fragments of bones, which were tapped on the surface of the stone surface, and in this way the stone tools were shaped. We also know that these bone fragments came from different animals, and that they were used by different human species like Homo sapiens and the Neanderthal man. And more importantly, we know that this retouching action leaves some specific marks, use marks, on the surface of the bone fragment, and we can study these microscopic marks to better understand the function of these tools. In Australia, the situation is different because archaeology is a much more younger discipline here in Australia. So although we have a lot of retouched lithic stone tools in archaeological context and in ethnographic reports, we actually still don't know how most of these tools were shaped. And with my PhD, with my research, that's what I'm trying to find out. So at the beginning of my PhD, I started to look about information about the retouching techniques in Australia, and I was very surprised to discover that there are a lot of references in the literature about boomerangs used to shape stone tools. And it was surprising because, in my mind, and I think in the mind of most of us, boomerangs are seen like as hunting tools. and that's it. But actually, it's not surprising at all, because as most of the Aboriginal traditional owners know, boomerangs have a big variety. First of all, they're not all returning boomerangs. Most of them actually, they don't come back when we throw them. And more importantly, they were not used only for hunting, but also for a lot of other functions like fighting, digging, even making music. I don't know if you hear the sound, but it's just beautiful. And so yes, it was not really surprising to read this literature evidence. So the next step, I wanted to have a close look to these boomerangs. So I went to 2 years ago now. I went to the Australian Museum in Sydney, and I just studied their boomerang collection. And actually, I published a paper last year about the results of this study, because in looking at the surface of some of these boomerangs, I actually identified some use marks that are very, very similar to the ones that we can identify on the bone retouchers, which we find in Europe. And it was a very nice discovery, and I wanted to know more. Of course, I also wanted to study more collections coming from different museums in other parts of Australia, especially in Queensland. But of course, because of all the pandemic situation, this was not possible. So I'm still waiting to get access to more of these collections. In the meantime, I designed an experiment to actually assess and understand how these how boomerangs could be used, could be functional to modify the shape of stone tools. So I had some
time is nearly up, Eva. You've got another 30 seconds.
Yeah, okay. So thanks to some Aboriginal craftsmen, I had these experimental boomerangs that then I used to try to shape stone tools. And so this is the movement that you do to shape the stone tools. And it was very interesting to notice that we got beautiful shaped stone tools using the boomerang, and on the boomerang surface we can identify the use marks that I saw on the ones on the boomerangs in the museum and on the
time is up now, Eva. If you could.
That's the last slide. That's the last slide. So yeah. Now I'm just, I am at the end of my PhD, but I want to know more about that. So in my future research, I hope I can find more collaboration. So if anyone wants to collaborate to try to investigate more this multi-purpose nature of the Aboriginal tool, of the Aboriginal tools, I would be very happy to. Thank you.
Our next speaker. And so the next presentation is by Henry Bartelet from James Cook University, and it'll be about the effect of coral bleaching impacts on the recreational value derived from the Great Barrier Reef. Do we have Henry? Henry's here. Henry, do you want to go ahead and share your slide, your PowerPoint or PDF? Or excellent.
Is this working?
Yeah, it's all good. We can see it. All right, go ahead. Thank you.
Yeah, so this is Henry here from Townsville, Northern Queensland, and today I will talk about tourism on the Great Barrier Reef, and specifically on the effect of coral bleaching on the recreational value that's derived from the Great Barrier Reef. So to start with the problem statement for this research, so human responses to changes in ecosystems are the basis for a wide range of complex feedbacks within social ecological systems. So understanding the ways in which adaptation by people mitigates the impacts of ecological change on human well-being is becoming increasingly important as the scale of human impacts on the environment increases. So in this research project, we tested the impact of climate-induced ecosystem change on the demand for and the satisfaction with recreation on coral reefs. So here in the Great Barrier Reef in Northern Queensland. And as many of you know, the GBR has been severely affected by coral bleaching in 2016, 2017, and 2020. So how did we study this? So we examined the effects of coral bleaching on tourism through the publicly available data on TripAdvisor. So this is one of the major review sites of tourism companies that probably many of you have used as well. So we included 41 coral reef tourism operators on the Great Barrier Reef for a total of 48,000 customer reviews. And that's between 2008 and 2021. And then we compared the average monthly visitor numbers and tour satisfaction ratings. And then we compared whether there was any change during or after the bleaching events in 2016 and 2017. And what we expected to find would be that that bleaching would have an impact on the visitor numbers, so that visitor numbers might decrease, and also the tour satisfaction might decrease. So that were our prior hypothesis. And to better understand any existing trends in satisfaction ratings, we also used the exploratory analysis of the written reviews. So you see an example here in the right. So the results, basically our first result is that we find that the ecosystem impacts from bleaching were associated with a reduction in the demand for recreation on the GBR. So here in the picture, you see the raw data is on the top panel, and then we filtered out the seasonality, and then we could see the trend. So basically we saw that from the year 2016, the first bleaching event, that there was a switch in the trend from growth in the number of reviews that went to a decrease. And then here in 2020, this one is from the COVID pandemic. So that was not part of our research. And also the data from the Great Barrier Reef Marine Park Authority, they showed the same trend. So we used that to cross-verify our findings. So this was in line with our hypothesis, but unexpectedly, we found that tourist satisfaction continued to increase throughout our sample period. So here you see that there's no effect in 2016 or 2017 on tourist satisfaction with their tours on the Great Barrier Reef. And we found evidence pointing to several aspects of the tour offerings that could have contributed to the increasing trend in satisfaction levels. For example, we find that the organization of the tours and the quality of the staff has increased. Knowledge about the ecosystem has increased, quality of food and beverages, and the quality price ratio, that these things could have caused this improvement. So there might be a number of explanations for the unexpected finding that visitor satisfaction did not decrease. So first, what I alluded to before is that there's potentially service substitution. So maybe the natural capital, so the quality of the coral reef declined, but actually the human capital, so the part of the other parts of the tour might have increased, so that might have compensated for that effect. It could also have been spatial relocation, so tourism companies might have changed their location of their tours. There might be delayed ecosystem effects, although we didn't find evidence for that in our data. And there might be shifting baselines. It could be that tourists don't have a baseline of what a healthy coral reef should look like. And the contribution of this study, theoretical, so we find evidence for the partial substitutability of natural capital by man-made capital. We also find valuable insights into the complex relationships between people and nature and the importance of better understanding adaptation effects in human societies and individuals. So feedbacks from ecosystems to the social system may be weaker than expected, and they might be unreliable if they are expected to drive corrective action that seeks to conserve and restore ecosystems and ecosystem service provision. We also have a few practical contributions. First of all, we have empirical evidence that the Great Barrier Reef is still a great tourist attraction, and that the quality of the tours and the experience might be better after the bleaching than before the bleaching. So I think this is a great finding now as well, that the international borders are reopening. We also have new insights into the role that ecosystem management authorities play in facilitating the delivery of recreation-based value. So the management authorities might play a role in facilitating adaptation by tourism operators, for example, by certification of tourism operator staff to become leading reef guides and ambassadors. That's something that's already happening. And authorities could also play a more active role in providing reliable and scientific information about the spatial characteristics of ecosystem damage and travel safety. So this is what I call managing the demand effect. So one thing we find in our study was that even areas that were not affected by bleaching, they saw a decrease in visitor numbers. So there was some kind of a mismatch between the information about the damage and the actual behavior of tourists. And this is also something we saw during the during the bushfire season, that locations that were 1,000 kilometers away from the bushfire were still seeing cancellations. So this is a role that authorities could play.
Henry, time is almost up. You have about 30 seconds left.
Yeah, I'm finished. Thank you so much.
Good, thank you, Henry.
You're there, Charles. Yeah, excellent. Go ahead and share your slides if
Yep.
okay.
Doing that right now. Yeah,
Excellent.
we're kind of invading from Griffith University, the
Yeah.
symposium. All right. Do you see it?
Yeah, all good. Go ahead and get started.
Great. Well, I'm going to talk to you about some mangroves and greenhouse gas emission today that is happening in North Queensland, where I did my study. So basically, mangroves, salt marsh, sea grasses, which lives at the interface between the ocean and the land, are very important ecosystems. And one of the many benefits that they give to us is that they store a lot of carbon. So they do buffer climate change. They have a great action. I think mangroves store more than rainforest, actually. But we don't really know how much greenhouse gas they emit. As you can see on the slide, there's a great variation, especially for methane flux, between like -1.1 to like 1,000 milligram of methane per day, which is a huge variation. So we don't really know what's happening there. We know that overall they do have negative climate forcing, which means they're buffering climate change. However, when they are disturbed, they release a lot more methane, especially when they lost a tidal connection with the salinity that it brings, which is supposed to be inhibiting methane emissions. So there's a lot of lack of information on the amount of methane they emit after these ecosystems have been restricted from their tidal connection. So what we did is that we went to the Bartelet catchment, which is one, well, I think that's the biggest catchment going to the Barrier Reef. And there's over there a lot of agriculture, a lot of sugarcane plantations. And they restricted a lot of the estuaries from the tidal connection to be able to build some freshwater reservoirs or for agricultural purposes. And some of this area looked like on the right at the bottom. So it's a lot of fresh water, sometimes with invasive paragraphs. What usually estuaries are mainly composed by salt marsh and mangroves. Now, what we did is a very simple study in a way that we just put PVC chambers onto the ground, and we took some air samples for 1 hour, and we used the accumulation of greenhouse gas emissions to calculate the greenhouse gas emissions. We also did a lot of studies on some of the factors that might influence these emissions, such as the carbon, nitrogen density, redox, which is a proxy for oxygen inside the soil. And so this is the result we got from last year. On the left, you got the tidal restricted, and you can especially see on the top part of that graph that tidal restricted are emitting a lot more methane than mangrove or salt marsh, between 20 to 160 times more. So, which is a lot. So that's kind of what we expected. But we also saw that they emitted much less nitrous oxide emission, which is another potent greenhouse gas. So that was a very interesting finding. And that the carbon emissions were kind of similar anyway. So the methane is really interesting. We also checked all the different soil physical chemical parameters, and we saw that with a statistical analysis, we could actually group the mangroves and salt marsh have the same characteristics, and they're very different from the tidal restricted, which forms another plot on the left. Especially that's the fact that the tidal restricted have a higher nitrogen and carbon density, which is known to positively influence the methane emissions. And at the contrary, tidally connected wetlands had a higher soil density and had higher oxygen in there. and methane emissions is a very anaerobic, very anoxic process. So that could kind of be used as indicators of methane or nitrous oxide emissions. And so as a global kind of summary of what we have in this system, so this is just about greenhouse gas emissions, but tidal restriction, what it does is that it will pass from a system which is emitting a bit of methane and a bit of nitrous oxides, especially storing a lot of carbon in there. And then we get a system which is emitting a lot of methane. And in terms of rehabilitation of the system, that brings a powerful tool, because now we see that if we actually restore this tidal connection, especially in areas which are not used anymore for agriculture, then we could actually buffer climate change in a way, and that provides a powerful tool to restore this ecosystem, because now we have these carbon credits happening, so we will actually use that as a financial incentive to restore these ecosystems. Now, this is one vision about the greenhouse gases. The tidal restricted wetlands also bring the really important ecosystem for biodiversity. There's a lot of meta-vertebrates living in there, so there's a bit of a more complex pictures to have here. But that was a really great finding, especially in the fact that we're trying to restore all of the Great Barrier Reef catchment to improve the resilience of the system. And yeah, as a take-home message, we can say that tidal restriction leads to a very high methane emissions, especially in the Burger King catchments, and that the tidal restoration is a powerful tool for climate mitigation. And we could use soil physical chemical parameters, such as carbon density, nitrogen density, or the amount of oxygen inside the soil. When we're going to do, if we do this restoration as indicator of restoration success. And that is it. Thanks for your attention.
Thank you.
Let's see if anyone has any questions.
Perfect timing. Perfect timing. Thank you. Is there anyone in the audience who'd like to ask a question, or perhaps provide a comment? Any feedback?
Can you hear me, Justina?
Yeah.
Yes.
Oh, good. Yeah.
Okay. Charles, there are many examples along the Queensland coast of tidal restriction. A classic example is in the Yeppoon area, where the Iwasaki tourist resort is, where 70 or more now, than probably 80 and 90 years ago, graziers produced bund walls to prevent the intrusion of the salt water from the mangrove systems, and then behind that planted pasture grasses so that they could graze cattle. And the question is, the cost of opening up that particular example would cost about $30 million. I'm just wondering, you know, there are cases where it is practicable. There are other cases where historically it will be quite challenging financially to address this. Any comment?
Absolutely. So we're mainly doing that in areas where we can work with farmers, especially in areas where they are not using the lands, because it is actually very complicated to do that everywhere. You can't do that everywhere. And there is also benefit from both sides. So I think it's
threats to threatened bird species in Queensland. You know, the big ones: land clearing, degradation, fragmentation, changes to fire regimes, invasive species. But there's also a few things going on in the background that, although they're less common, can also have a significant impact on threatened species. And one of those, I believe, is intersex in birds. Now, there's a wide spectrum of intersex. So to be really clear for what I'm talking to you about today, I'm saying that intersex is where you have genetic females that show up with a male phenotype. So this is complete with male plumage, testes, and genetic males that show up with a female phenotype. So this is where the secondary sex characteristics are, quite opposite to what they show up as. So sex reversals can show up at different times in a bird's life. Adult females can masculinize in the absence of estrogen, and that's caused by a diseased or degenerative ovary. Now, in monomorphic species, this can go quite unnoticed, but in species where there's quite a difference between the males and the females, it can be quite obvious, like in this mandarin duck here. But the kind of sex reversal or intersex that I'm concerned about is one that happens in ovo, so it happens in the egg. Now, this is quite fascinating. So in the maternal environment, we don't know a lot about it with wild birds, but we can look at lab studies to tell us about how this happens and suggest the prevalence of it. So lab-based trials have shown that, yeah, these large elevated levels of corticosterone or endocrine disrupting substances in the female's environment, anywhere up to 2 hours before she lays an egg, can actually be laid down into the yolk and the albumin and interrupt that natural cascade of events that happens after fertilization, but before the development of the gonads, and permanently reprogram the adult phenotype of that offspring. So you might wonder why this is a problem. Now, in birds, lab studies have shown that there can be a great miscommunication between the brain and the parts of the and the gonads, and interrupt essential communication paths that are really essential in the beginning of breeding season for things like the development of follicles, the budding of follicles, enlargement of testes, pair bonding. Other problems with sex reversed individuals is that in sex reversed females, their sperm can be misshapen or lack motility. So this can lead to low fertility rates or just a complete inability to fertilize an egg. Now, another consideration is that in birds, females are the heterogomatic sex, meaning they carry both the Z and the W sex chromosome. It's a different system to us. And in males, they have 2 Z chromosomes. So by the very nature, I guess, of their genetic makeup, a sex reversed male can only produce male offspring. Now, in small or otherwise, I guess, at threat populations, where every pairing is critical, these individuals can actually play quite a major part in reducing the operational sex ratio. Now, we know that the sex ratio of a lot of threatened bird species is skewed, usually to male, but what we don't know is if intersex birds are playing a part in either low productive output or in perceived sex skews. So that's something that, as part of my thesis, I aim to look at. I wanted to know, I'm a population biologist in a captive sense from the zoo industry, and I wanted to look at the incidence of intersex in wild birds to sort of predict, I guess, the likelihood that this could be happening in threatened bird species. So I chose 5 local species and across 4 orders, and I dissected over 460 of them. I took, with the dissections, I took a liver sample for DNA analysis. I also observed and measured and photographed the gonads and took morphological measurements. I then extracted the DNA and used a single primer set in PCR analysis. When I looked at these on a gel, they revealed a single...
Time is almost up, Clancy. Thank
you. Sorry, that showed a single bar for males and 2 bars for females. So that using this information, I was able to cross-check my information and look at whether I had intersex individuals. So I found intersex individuals in every species I looked at. Most of these individuals were genetic females with testes, and there was also a kookaburra that was genetic male that was in the middle of egg production.
My goodness.
Which is quite amazing. Now, there's very little studies existing on intersex in birds, but in other species, prevalence sits at around 10% in fish and frogs. Bearded dragons, recent research has shown up to 8 to 22% in southern Queensland's population. In mammals and birds with more traditional genetic sex determination, you might expect much lower levels, and there are. In mammals, high rates are considered in farmed pigs at about 1%. So where do the birds fit in? Laughing cookie, they're somewhere in between 4%, over 5% in rainbows, and just over 6% in crested pigeons and scaly-breasted lorikeets. So I think overall, going back to the aims of my study, I would say that there is a chance that intersex is playing a part in threatened species demise, and that this is something that we need to watch, especially in polluted environments where the incidence can be tenfold. So I'd like to thank everyone for your attention today, and for the following people and institutions for their support.
All right.
Excellent. We can see everything. Looks good.
Go from the beginning. So you can see those slides there.
Yeah.
Yes, all good. I think we're looking at your like the toolbars that you've got to show the PowerPoint. So you can perhaps move your screen sharing to the screen where your actual slides are, or that's the one.
That one.
Couldn't find the words to describe this.
That's right. There you go. Okay,
That's
I'll
great.
move you over there. No
Perfect.
worries. Perfect. All right. Yeah, my name is Gabrielle, and I am speaking from Turrbal and Yagara country today, Meanjin, Brisbane. I'd like to pay my respects to elders past, present, and emerging. So today I'm going to be presenting some of my PhD research where I investigated land use change and the spread of invasive species in Queensland. So I'm sure many of us have seen these headlines floating around within the last 10 years or so, highlighting Queensland's devastating rates of land clearing. We really are up there with some of the highest around the world, and this has largely been for the expansion of cropping and grazing. So this has had huge impacts for our fauna and flora. This paper here by April Reside shows that for 29 species that are listed endangered in Queensland, the loss of woody vegetation habitat has been listed as a key threatening process. So land use change and clearing also has several indirect impacts, including changes to fire regimes, nutrient and hydrological cycling, dispersal limitations, and the spread of invasive species. So during my PhD, I spent a lot of time within these cleared and modified landscapes of Queensland, and really got some firsthand experience of how land clearing was impacting plant communities. And one experience that really sits with me is in 2018, when I went out to resurvey some sites that my supervisor, Rod, had previously surveyed the vegetation at in 1996. So my PhD was mainly focused on this invasive grass, Indian couch. So Indian couch was and is rapidly spreading throughout Queensland, and my PhD was looking at documenting its spread, impact, and management. So when I went to look at these sites, I was mainly focused on Indian couch and how its cover had changed. But I very quickly realized, throughout the survey, that this was not just a story about couch, and it was really about the whole landscape and land use change, and how these invasive species were spreading within these modified landscapes. So I shifted the focus of the survey. So just to provide a little bit of context, in 1996, my supervisor, Rod, pictured here, went out to survey 200 sites in this focus area of central Queensland to document the flora within dominant grassy and grassy woodland ecosystems, and to provide advice on their conservation, because they were recognized as increasingly under threat with crops and grazing. So he was also looking at invaded cover, particularly bufflegrass and Parthenium weed. So bufflegrass is a perennial grass that's commonly sown for pasture, and Parthenium is an annual herb shrub that is toxic to stock. He wasn't really looking at Indian cooch in '96, but thankfully recorded the cover of all species. So I could also look at this species. Okay, so in 1996, when Rod established these sites, they were all within native dominant, they all had a native dominant understory and were used for grazing. I wanted to know what was going on in 2018, how land use had changed and how had the cover and occupancy of these focal invaders changed. So to answer this first question, I used vegetation mapping, land use mapping and on-ground surveys. And what I found was that 164 of the sites were still used for grazing, 7 were within conservation reserves, 36 had been converted to cropping. And what else I found is that clearing of remnant vegetation had occurred at 17 of these sites. So, and this was 15 associated with grazing land uses and 2 for cropping. So overall, there were 51 sites where land use had either changed or intensified. So to answer question 2, I resurveyed invader cover using the same methods that Rod did in '96 across 54 of the sites that were randomly chosen. And what I found was that all invaders had increased in cover and occupancy, particularly for Indian cooch and bufflegrass, and particularly for Indian cooch, as you can see from that graph there. So what this survey really highlighted for me was that the direct and indirect impacts of land use change. So over the 23 years, we found that 51 sites experienced some level of land use change or intensification, and that increased invasion was occurring across several of the sites. So in a recent paper that we published, we used this site data as well as some landscape scale data to argue that the spread of these invaders in this region is an example of invasion debt incurred from an intense history of land use change in the region. So what we mean by invasion debt here is that the spread of these invaders is not necessarily a direct impact of land use change, but is ongoing, or is occurring over longer time frames and will likely be ongoing. What we stress is that when we consider these indirect ongoing impacts of land use change, the consequences for biodiversity are likely far greater than if we just considered direct impacts alone. So we really do have work to do in Queensland to manage and reduce these impacts of land use change. Some key things that we can do is to protect our remnant vegetation before it becomes threatened.
Time is almost up, Gabrielle.
Thank you. We can restore ecosystems. Many of these heavily cleared landscapes do have high potential for restoration. We can manage our grazing pressure. So some of these invaders like Indian cooch and Parthenium are facilitated by high grazing pressure. And for all of these things to happen, we really need to develop strategies to balance cattle, crops, and conservation. And a promising way of doing this is evidence-based programs which incentivize the protection of biodiversity on private land. So thanks so much for listening, and please, shout out if you have any questions.
Ryu, do we have you? I'm just looking through the participants. I'm pretty sure Ryu was here, but in the morning anyway. I'm just going to double check. Let's give it maybe 1 more minute. I'm just checking. Ryu isn't here in the list of participants at the moment. Bear with me just 1 moment, everyone. Possibly maybe like internet issues or something. I don't know. Let's wait a couple of seconds. Don't see Ryu at all. Peter, am I looking properly? Can you see Ryu Lippman in the list of participants? Oh, there we go. There's Ryu.
Hi, sorry, Yeah, I'm back again.
You're back again?
The internet's
Excellent.
been fine all day, and then suddenly,
Oh, good.
you
Okay,
know,
we're fine. It's only
how
11
knows
past.
when the time comes. Am I back?
You're back. You are
Okay.
breaking a bit, but let's go ahead. Let's try.
Okay.
Let's try.
Can
Okay.
you see my screen?
You just started sharing. We don't see anything.
Should I try turning off my video?
I mean, this is all good. We can, yeah. Yes, please switch off your video. We can see your screen. Let's give it a go. Yeah. We still can't hear you, I'm afraid. Can you actually? Okay.
If I stop my video.
Sorry, we can't hear anything, unfortunately.
I could hear him. So there's something awry with links, because I'm listening to him and hearing.
Yeah, I can hear too.
Okay, yeah, okay, all right, let's keep going then.
All right, I'm not sure what to do. Can you hear me? Or is it all broken up? Can't hear it all.
It sounds like others can hear you, so it might be on my end. So just keep going, and then I'll sort it out on my end.
Gone again. He's gone again.
I think we lost him again. Yeah. I propose that. Yeah. I mean, shall we just wait, and then we'll have the next speaker in the next session. Sorry, for the next talk, and yeah, let's just take a bit of a break. Oh, that's a shame.
See if he comes back. Yep.
See if he comes back.
They log back into Zoom. Sometimes it improves the connection.
Yeah, go out.
Yeah, good point. Yeah.
I still don't see. Yeah, I still don't see Ria in my list.
I think you've definitely got transmission challenges there.
Yep, that's right.
Looks as though Alexandra is ready.
Yeah, if you wanted to, I'm happy to have a go now.
Yep.
Yeah, all right, we might as well, I guess. All right. Okay, everyone, apologies there for the technical issues. So we've got Alexandra Matteson from University of Queensland talking about the identification of novel peptides in Cavendish banana during Fusarium wilt infection.
Yes, thank you so much. Hopefully we don't have any more tech issues, but my name is Alex. I'm doing my PhD at the University of Queensland, actually with the Integrative Legume Research Group. But I'm not going to be talking about legumes today. I'm talking about a new project that's going to be happening this year thanks to the funding from the Royal Society of Queensland. If my slides cooperate, there we go. I'm actually going to be talking about bananas today. So I love bananas. I have them in my cereal. But I think there's a dozen reasons why everyone else should also be excited by bananas. And that's including their nutritional and economic value to us. So obviously they're a very healthy snack that everyone can enjoy. But in addition to that, in Australia, it's a $596 million industry. And 94% of that industry actually happens right here in Queensland, which is really exciting. So something you might not know about bananas is the wild varieties actually have seeds in them, which is very inconvenient for us who are trying to eat them. So you'll actually find that the Cavendish and ladyfinger bananas are actually sterile crosses of bananas, so they don't have seeds. So when we're farming bananas and we don't want them to have seeds, we actually need to do something called clonal propagation. So we don't have seeds to reproduce the bananas. So there are a few techniques where we basically make clones of the same banana. And so this can be through tissue culture. This looks very sciencey, or it can also be through something called suckering technique. And so a farm of bananas is basically a field full of the same or clones of the same banana. Now, this is a little bit stressful for the system, because when a disease happens, if a disease should occur, that means that all of those bananas are going to have the same genetic code, and they're going to all be affected by that disease. There's no chance that there's going to be resistant varieties in that field. So one of the diseases that we're a little bit stressed out about is called fusarium wilt. Now, fusarium wilt is a fungus that comes through the soil and infects the banana's roots. And what it does, it actually plugs up, it affects the vascular system of the bananas and stops water being able to get up through the plant. And so you can see this middle picture here, that's called skirting, because it looks like a skirt on the banana. It's called skirting when the leaves basically fall down because they can't get any water. And you can see in this picture on the right, those dark rings are all fusarium mycelium growing in there. So remembering that all of the bananas are clones of each other, they are all susceptible to this disease. So what are we doing to control it? Unfortunately, there are no chemical or biological control methods currently available to help control this disease. So farmers are relying on really good hygiene and quarantine practices. So that's if there's any chance that there is a disease there, it's quarantined and blocked off so that the rest of the population doesn't get infected. And that might be, if you've noticed, you can't just go to Bunnings and buy a little banana plant, and that's because of all of the rules around the quarantining and transport of bananas in Australia, because we're very cautious of this disease. So obviously, this isn't a great situation to be in. It can be very stressful for farmers who are, have banana crops should this disease appear. So obviously the research, the scientific community is putting a lot of research into trying to find alternatives to try and control this disease. And the more we understand about the bananas and the disease itself and what's going on a molecular level when they interact, the more likely we are to be able to come up with an alternative control method. And so that's when I would like to introduce a really cool little peptide called clepeptides. So clepeptides are a plant peptide. It's a family of peptides, and they're really small, and they're a signaling molecule, so they act kind of like hormones. And this image shows that they are involved in a lot of different regulatory mechanisms in the plant, and also a lot of interactions with microbes. Now, you may have noticed that this is not a picture of a banana. This is actually an image of a bean seedling. So I've been working on these peptides in my PhD with beans so far, and taking a look at their potential role when the beans get a pathogen, because we know that they're involved in so many other microbe interaction pathways. So naturally, the next step is to see if we can apply this to bananas. The project that I'll be working on this year will involve looking at bananas that are infected with fusarium wilt, and taking a look at the potential role of clepeptides in the plant when this happens. So obviously, firstly, we need to find the clepeptides in the bananas. They actually haven't been effectively screened for clepeptides yet, so that's our first step. And then we also have some really great samples and data collected through Elizabeth Aitken's research group of bananas that are resistant and susceptible and that have been infected and not infected with this disease. And so then we can take a look.
Up, Alex.
Okay, thank you. And then we can take a look at what role the clepeptides are having in that interaction. So a big thank you to the Royal Society of Queensland, because this really wouldn't be possible without the funding. It wouldn't be happening at all. And I'm really thankful to have a really great team of people working with me to get this done. I'm happy to answer any questions if you're.
It's going to be a bit shortened, but I'll give it a go.
Screen and
Okay,
just.
so I'm looking at optimizing facility location for coral aquaculture production. So coral reefs are the most biologically diverse, sensitive. And the world's coral reefs maintained and restored by active reef restoration.
I think he dropped out again.
Yeah.
Oh, sorry, how frustrating. Okay, I would suggest that we try one more time at the end of the second session, perhaps. And if we're continuing having these difficulties, perhaps I could offer Ryu to record, like pre-record his talk, not pre anymore, but record it. And then I'll share it with everyone and we'll upload it to YouTube so that we can still give him the chance to deliver the talk. What do we think?
That's good. Yes.
Yep.
Okay. All right. Apologies, everyone. And Ryu, if you're there, sorry. I wish.
Sorry. Yeah, it seems to be when I share my screen, my internet can't handle it. Sorry.
So what we might do. So did you just hear what I was saying? So what we might do is try one more time at the end of the second session. Why don't I share your slides? I've got them, right? Because you sent me a copy. You can talk.
Yep.
We'll see if that works. And if it doesn't, why don't you record it in your own spare time and then we'll share it kind of electronically with everyone.
Okay. All right. Thanks. Sorry about that.
No. I'm sorry it's happened to you. The joys of doing everything online in the pandemic, everybody.
Yeah.
But thank you all for everyone's attention and presence in the first session. Let's take a quick break, grab a coffee, grab a cup of tea, whatever you want, and I will see you back at 10:40. Thanks everyone again.
Thank you.
Ross, that went really well. If you're still there. I'm just saying it went really well. And
yes, very exciting. I think it's been extremely successful so far and very motivating. Good stuff.
Absolutely. I mean, the interesting thing to me is that these young people felt that it was worth doing with the Royal Society, and that is what we're after. And so we can build on that, I'm sure.
See, I think also, while we've had overhead costs that we're trying to accumulate from membership, at their stage in their career, we can offer associate memberships at a third of the cost, or $30, or the old price, $35, just to get them involved, because this is a gateway for their publications.
Yep, and also for extending to friends and whatever, colleagues. Because if the word goes out, gee, this went well, next time we run it, we will get another tranche, and so on. So I think if Justina and Cole and others who've been involved in organising it can cope, we might look to run them more times, even 3 times a year.
No, it's very exciting and very worthwhile. And it's enlightening in terms of the advances in methodology and the intricacies of the questions being asked and the sort of outcomes of the work. It's really worthwhile.
And I look back to my own career and my own early public talks and so on. These people are so confident in their presentation. It's wonderful how somehow we've gone from the nervousness of early career presentations to being so confident and capable.
Yes,
it's excellent.
I was looking at last night. I looked back to my first sort of refereed publication in 1974, and basically I was just thinking then, yeah. Compared to some of the presentations we've seen today, my must have been fairly shaky. Yeah, although I must admit I was a bit of a confident guy at that stage anyway. Yeah.
Yes, indeed. I mean, it's a journey that is, in a sense, being truncated through this internet connection stuff. You don't have to go to conferences and travel the world to have the joy of being able to present your work in a comfortable and rewarding manner. And I think that's something that, we can go into that space.
It's a very good point. Yeah. Yeah, no, it's very encouraging and very worthwhile.
Yeah.
So, and Justina's done a marvellous job in organising.
I've got to remind Justina that we have met and we don't need to meet for a coffee for the first time. She and I had a long and enjoyable conversation at the government house. And the last occasion we were all there.
Okay.
So, so I'm going to remind her. She and Julian and I had a great talk. Julian was whisked off to meet the governor, and so Justina and I chatted for most of the evening, actually.
Right, no, that's good.
Yeah.
No, she's done an excellent job.
I'm back, Peter. I'm so sorry.
No, don't worry. I just realised as well.
I thought
I'd forgotten.
Makes sense. Yeah, that makes. I remember. I think we talked about anthropology and various other things.
Yes,
and I just
we had
moved out
great.
to something. Oh, that's hilarious.
And
well, I'm just reading her.
And of course, and I was keeping an eye on you both and thinking I better separate you because you might be bored out of your wits with each other.
No,
it went really
absolutely not. We look forward to more conversations.
Jeff, your note about free membership for a year, I support. Yeah.
Yes, that'd be good. That'd be a really good way.
Would you like to offer that, Ross, to the meeting when we reconvene?
Yes, I would.
I find this very interesting. I can't even find the time to get up and make coffie for myself. It's
I was making a cup of coffee when Peter got me involved in discussion. but basically, at
the University of the Sunshine Coast, and yes, transcriptomic analysis of tissue regeneration in crayfish identifies stem cell and myogenesis genes, which are potential tools to develop cultured crustacean meat. So this project is part of an ongoing endeavour to advance cultured crustacean meat here in Queensland. So ultimately, we're aiming for prawn cocktails and lobster mornayes without the need to farm or fish. So before I touch on the why behind cultured meat, I'll briefly cover what it is. So basically, this emerging technology involves growing real meat tissues from animal stem cells rather than whole living animals. So inside food grade facilities, these stem cells are made to proliferate and then differentiate into just the meat tissues. Many prototypes have already been made, with some already approved and sold in Singapore. So how do scientists grow meat in vitro? Well, this can be done via two broad approaches. Firstly, by providing stem cells the right cues to proliferate and then differentiate into meat-relevant cells through their culture medium or scaffold materials. Or it can be done via overexpression of particular key regulator genes, such as MyoD, which can initiate the myogenic program in many stem cell types in vertebrates. So why cultured meat? Well, this centres around it being an alternative to conventionally produced meat, because it takes a lot to grow animals just to produce the products that we eat. They take up proportionately more land and resources than they contribute to our protein supply. And because we need to raise and slaughter so many animals, current meat production is the leading cause of deforestation, global warming, pollution, public health threats such as zoonotic pandemics, and animal suffering on a massive scale. And as the world population increases, it stands to reason that finding alternative, less destructive methods to produce meat are needed. So why crustaceans? Well, for one, they make really popular foods, particularly here in Queensland. And their current production also involves numerous issues that warrant finding alternative sources. So we have overfishing, pollution, microplastic contamination, and constant disease threats, which are just some of these. And here in Queensland, warming oceans and climate change are increasing threats to existing farming and fishing models. A number of more sustainable onshore farming systems do aim to address many of these issues. However, they are costly and unlikely to meet growing demand alone. So clearly additional supply sources are needed. And crustaceans are currently underrepresented in the field of cultured meat. And this is because there aren't any crustacean cell lines available. There are few established cell culture protocols, and there's very little known about what molecular mechanisms drive crustacean stem cell growth and myogenesis. So there's a clear knowledge gap here. And not only can this research support the development of an exciting and sustainable industry here in Queensland, advancing our understanding of crustacean cell culture and developing new cellular and molecular tools will also help improve our existing industries, particularly in terms of disease resistance research and more sustainable productivity. So my research focuses on the regenerating limbs of the Australian red claw crayfish. Like all decapods, they can eject their limbs and at a preformed plane and regrow them. And once the limb has been ejected, a blastema forms, and this undergoes epimorphic regeneration, where you have undifferentiated stem cells replicating, and then they eventually differentiate into mostly muscle tissue, which is of course the main constituent of meat. So we hypothesized that the limb regeneration process should involve all the genes and the cells required to replicate myogenesis in vitro. We decided that an RNA-seq analysis across the process could help us identify our target genes and highlight where they are most active. So this will hopefully shed light on the best tissues to use for cultured meat experiments. And using regenerating limbs is a more humane approach, as it allows us to access these tissues without any permanent impact on the animals. So the methods are the RNA was sequenced and assembled into a transcriptome using Trinity software. The crayfish homologs were identified using homology searches, domain feature checks, and phylogeny. And expression was quantified to give us relative expression patterns of the genes across the regeneration process. So here are the relevant growth, muscle, and stem cell related genes that we putatively identified. We found that genes from each of those categories seem to be most active simultaneously in stages 2 and 4, which actually aligns with 2 known growth phases in decapod limb regeneration. And these tissues potentially harbour cells that are more primed to proliferate and differentiate into muscle, and could be ideal candidates for starting cell cultures for cultured meat experiments. So of particular interest to me also were the genes Twist and Nautilus. And these are the fruit fly versions of the master muscle regulator MyoD. My analysis found that these genes were expressed during different growth peaks, which is potentially interesting. To validate this pattern and my other findings, I'm currently conducting qPCR experiments to confirm actual relative expression of my identified genes. And once completed, we should have a better understanding of the best tissues to use in cell culture experiments.
Time is almost up, Lisa.
Okay. So ultimately, we like to find the ideal conditions for growing crustacean stem cells and efficient ways to turn them into crustacean meat. So I believe this research has the potential to improve our food system, but also boost local industry. So currently the vast majority of products consumed here come from unsustainable Asian imports. So developing a cultured crustacean meat industry could provide a good alternative to that. A supply that is local, safer, more ethical, more sustainable and ocean friendly, and more future ready. And furthermore, the tools are translatable.
Now, time's up now, Lisa.
Great, thank you.
Innovation. This is Jodie Neal, who's the lead breeder in our team. And we have a really big team across the country. We're based at Nambour, but we're a national program. So we breed for new varieties for 3 different regions. We've got the subtropical region, which is Bundaberg and on the Sunshine Coast, and that's your winter strawberries. Temperate, which is in the southern states and also around Stanthorpe, and that's the fruit that you're getting now in summer. And then also the Mediterranean region near Perth. So our varieties have been performing really well. We have a new subtropical variety called Susie, which is inspired by Susie O'Neill, which should be available soon in supermarkets. And it takes around 5 to 6 years to breed for new varieties that have a good combination of traits for consumers and also traits that make it profitable for our hardworking farmers. So as part of the breeding process, we assess our varieties for susceptibility to different diseases. And unfortunately in strawberries, there's lots of fungicide sprays that are applied to control the diseases. But as we all know, there's an industry, environmental and societal push to move away from chemical sprays into something more sustainable. So one of the diseases that affects strawberries is powdery mildew, and this is Apollo. So he is here assessing each individual plant 3 times in a season on a scale of 1 to 5 for both the leaves and the fruit, with 1 being okay and 5 being really symptomatic. So you can see here, this is the fungus and it affects the fruit and the leaves. The leaves curl up, which impacts photosynthesis and therefore yield. This is one of our really successful varieties, Red Rhapsody, and that gets a score of around 2 or 3. So with the scale going up to 5, it can get much, much worse than this. And this is one of our non-commercial selections, which is powdery mildew resistant, but it's not commercial, it's too small and too soft. So, but that's good news because it means that we have genetic resistance in our germplasm, and it means that we can breed for resistance. So we can cross a plant that has powdery mildew resistance with a plant that has high yield and large fruit and other good quality characteristics to hopefully produce offspring that have the best of both worlds. But testing all of those plants in a disease screening trial is time consuming and expensive. So that's why we're looking at DNA data to screen for resistance instead. So there are many differences in the genetics among plants, but only some of them are due to differences in susceptibility to powdery mildew. And I've been using 2 different genomic methods to find these genetic differences. We've got genome-wide association study, or GWAS, and genomic selection. So in GWAS, this works well for simple traits that are controlled by a few genes of large effect. And this method looks at each marker individually, and the results are used in micro-assisted selection and for diagnostics. In genomic selection, this is better for traits that are more complex, that have many genes that are controlling it, all of small effect. And this method looks at all of the markers or the specific DNA locations simultaneously. And genomic selection is used in predicting performance and making selections. And so both methods combine trait performance data with genetic marker data. So for GWAS, we look at the DNA for plants with powdery mildew versus those without powdery mildew. And we have an algorithm that tests each marker individually to find alleles, or the A, T, C, and G, that are different between the two different groups. So here we have a difference where there's a C allele for those plants with powdery mildew and T for plants without powdery mildew. So we explored this a little bit more. For this genetic marker, I compared each plant's alleles, the C's and the T's, with its mean leaf score, so mean over the 3 time periods. Remember, 1 is good and 5 is bad. So here, this allelic state of CC corresponds with more severe symptoms versus TT, which is not as severe, it's more resistant. So ideally, we would prefer to have plants that have TT rather than CC because it's linked with resistance. So that's GWAS. Now for genomic selection. So this method combines the trait performance data with the genetic marker data to construct a statistical model. So it's like looking at a pattern across all of the alleles, all of the genetic markers to see how they correspond with the trait. So we then apply the statistical model to the DNA information of a testing population to predict their performance using only their genetic data. So the prediction accuracy of the model is the correlation between the predicted performance and the known performance. And we've developed some moderately accurate prediction models for powdery mildew severity in leaves and fruit. So correlation of 0.52 in leaves and 0.59 in fruit. So as we get more data, both the trait performance data and the genetic data, we can add this in and construct a more statistically robust and accurate model. So what does this mean? Well, first we need to validate our results and our models to make sure that they hold true across different environments. And then we can examine the DNA really early in the life cycle of a plant. So just after germination, when a plant's got its very first leaf, we can extract DNA from that leaf. And we can either look at the specific genetic markers like this one and select plants that have T's rather than C's. Or if we have all of the genetic marker data, we can use genomic selection to predict their performance. So this means that we can identify the best plants really quickly and discard the poor performers and instead focus just on the winners. And then we can combine our powdery mildew genomic selection models with other models that predict for yield and fruit size and sweetness and things like that.
Time is almost up, Katie.
Thank you, Pam. I'm done.
Thank you so much. Go ahead and get started, please.
Excellent, thank you. Hi everyone, thank you for the introduction, Justina, and thank you to the Royal Society of Queensland for the opportunity to present here today and the membership, which is an amazing bonus. As said, my name is Cynthia and I'm a PhD candidate in biological anthropology with the School of Archaeology and Anthropology at the Australian National University. And today I'll be presenting findings from a qualitative study that we've undertaken as part of my PhD research into an association between cyclones and perinatal health. Environmental disasters present both objective hardship and subjective distress to pregnant women. Cyclone disasters have been associated with an increased risk of pregnancy complications such as gestational diabetes and an increased risk of adverse birth outcomes such as preterm or low birth weight births. In addition to vulnerabilities that can be brought on by the roles and responsibilities of mothers within a family unit, a mother's own individual perception of events provide insight into how she handles situations and what increases her vulnerability or strengthens her resilience. Despite all of this, no studies have looked into the individual experiences of mothers pregnant during cyclones in Queensland. So I aimed to address this gap by collecting and analysing maternal accounts using semi-structured interviews. Mothers could participate in the study if they had experienced a Queensland cyclone during pregnancy. Queensland was selected as the study region due to its large population, which mostly resides along its coastlines, its variable geography, and vulnerability to seasonal cyclones, of course. Interviews were conducted from June to July 2020 and were semi-structured in that as mothers, prompts were provided to mothers if needed during the interview itself. Transcripts from interviews were coded and assigned themes using the methods recommended by Braun and Clark. I drafted transcripts for the 12 mothers interviewed and applied codes. These codes were validated by a second coder, and themes were drafted and discussed by the research team. 3 themes were agreed upon based on the validated codes and sub-themes. Themes were aimed to represent commonalities and differences in experiences across the mothers interviewed. This included drivers of risk perception, awareness and evaluation of cycle risk communication, and maternal appraisal of the overall event. I'll now go into more detail on each theme and provide example quotes from the transcripts themselves. The first theme was around drivers of risk perception that were mentioned. These would include sources of resilience or vulnerability at the time of the cyclone, such as support from their immediate community, their past experiences, knowledge of emergency protocols, and how being pregnant influenced the experience. In this quote, the mother provides an example of support received, particularly when limited by her pregnancy, when she says, "I think I probably felt less prepared because of the physical incapacities. I couldn't do a lot of the things that I would ordinarily have done for cyclone preparation. So I relied heavily upon my husband to be able to do all of that." Partners, neighbors, and grandparents were mentioned by all mothers, particularly in the preparation and recovery period, providing support, looking after other children, driving them to the hospital, in this case, preparing for the storm by lifting heavier objects. In this quote, past experiences and quintessential Australian attitudes to the cyclone were mentioned by a mother as influencing her risk perception. As she stated, "I guess my family's probably more easygoing people and more typical, Aussie people, should be all right," and all of that. Obviously, living in the area as well and having it drilled into you that you need to be prepared definitely takes an element of fear away from it. The second theme involves the mother's awareness and evaluation of risk communication and the role this played in her preparation for the cyclone and recovery afterwards. This could include whether she had a negative or positive interaction with the available risk communication and provided insight into what resources were used at the time. In this quote, a mother summarized the resources she relied on and those she was less likely to use and why. We tend to go to BOM because it's one of the ones that everyone goes to. When you listen to the news, they tend to sensationalize a lot of stuff. So we didn't really even listen to the news. We just kept going to BOM every few hours when it sort of refreshed. And we just kept an eye on it there and gone, right, well, it's coming this way. And do we look at the tracking map and look at what category it's going to be when it hits? Sensationalization of the cyclone in the news was something brought up by a number of mothers, but this was based on them having had past experiences of cyclones to identify when a report was being sensationalist. The last theme involved a mother's appraisal or reflection on the overall event, providing her personal insights such as emotions felt, key takeaways, and anything that would be done differently in the case of a similar event. In this quote, despite the mother having an overall positive reflection on the events, she would not want to repeat it, as she said, "I think it was a good experience, but I think next time," and my husband said this, that next time if a cyclone comes, we'll just be getting in a car and driving out west and just let the house be and not go through it. In the second quote, a different mother provided more information around why she would consider the overall experience in a positive way. Her experience had included a pregnancy loss at the time of the cyclone, and this was a reflection on both the cyclone and the weight of that loss. I can see the lighter side of it, if that makes sense. Like I can see the silver lining, I suppose. Like if it makes you a bit stronger, and a bit more resilient to stuff. And you seem to be able to get over stuff a bit better if you can get over that hump. Findings highlighted the importance of future work exploring a more diverse sample to capture the experiences of more marginalized mothers who may not have access to similar supports, local knowledge, and past experiences as the mothers interviewed. Despite the interview sample being small and homogeneous, it provided invaluable insight into what mothers consider when pregnant during cyclones in Queensland. Above all, it also highlighted multiple sources of resilience that strengthened a mother's feeling of preparedness when faced with a cyclone during her pregnancy, such as through the support of her immediate community. A lot of literature highlights important sources of vulnerability when faced with a disaster, but we are now able to add sources of resilience that can support mothers and those supporting them when faced with a pregnancy during a cyclone.
Time is up now, Cynthia.
Thank you.
It's declined pretty substantially, particularly in the last 30 or 40 years. So in the last few decades, we've lost at least two previously known populations of the bettong. So from down here in the Cohen Range, just north of Townsville, and Mount Windsor, northwest of Port Douglas. The last records from both of those sites were in 2003. And then there's this little population in Ravenshoe, which was lost sometime before that. So the two remaining populations, there's quite a large healthy population here in the Lamb Range, and then a small population a bit further north at Mount Spurgeon that seems to still be declining. The major threats and drivers for these declines are thought to be fire regime, so namely a lack of fire at some of these sites, which has allowed rainforest encroachment. So the bettong requires pretty open grassy habitat for nesting and for feeding. So with a lack of fire, we lose a lot of this habitat. And potentially the impact of predators, so particularly feral cats. And that's another aspect of my project, which I won't touch on today. But if you're interested, feel free to get in touch. My study sites are within this Lamb Range population, so specifically at Davies Creek, which is on Bulli country. And we wanted to look at how bettongs respond to fire in this stable population. If my slides want to respond. Cool, so that's the overall research question. How do northern bettongs respond to fire? And we're looking at this at the population level and at the individual level. So we performed marker recapture surveys to estimate population density before and after fire. And then while we had the bettongs in hand, we took a number of measurements. So things like body condition, weight, reproductive status, things like that. And then we GPS collared 20 bettongs over 2 field seasons. And we were able to track their movements and home range use before, during and after fire within their home range, and then analyse this against fire maps to see how different fire intensities across the landscape impacted their habitat use. So at the population level, basically good news all around. The population density across these fires, there was no change, and these densities are also in line with previous work at this study site. So this shows a stable population over at least the last 30 years or so, and no changes to anything like reproductive rates. Pretty much 95% of all females we caught were carrying pouch young. Body condition didn't change, and BMI, there were no change either, so body mass index. So basically the bettongs at a population level were healthy and maintaining stable density. At an individual level, we mapped their home ranges from GPS collars and plotted these on, so this is fire intensity maps, where the green areas were unburnt patches up through to the red areas, which are high intensity fire. Basically from this plot, this is overall home range, and we found no consistent trends, no significant differences between pre and post fire home ranges in bettongs. And so we drilled into this a bit further and separated each bettong's home range into basically nesting and foraging areas based on the timing of the GPS fixes and movement patterns and things like that. And this is bettong nesting areas. So we found the foraging area pretty much resembled the overall home range. But nesting areas, we had a significantly higher amount of unburned habitat in their nesting area post-fire compared to pre-fire. So again, it's a little bit inconsistent, and this, looking at all these plots, doesn't show it all that clearly, but this one is quite a good example. This individual, you can see the solid line being its pre-fire home range, and a lot of that was burnt and burnt at sort of moderate intensities, whereas the post-fire nesting range incorporated a lot more unburned habitat. So bettongs nest in predominantly in grass clumps. So basically it makes sense, unburnt habitat, they'll shift where they're nesting to find those grassy clumps. And this is really, I guess, a good example of the patchiness of these burns and how important that is for bettongs. Whereas the overall home range didn't shift. And I think this shows a really good adaptation of bettongs to sort of low to medium intensity fires, because their food sources are predominantly underground. So they feed mostly on fungi, grass roots and tubers and things like that. So as long as fires are of a low enough intensity, these food sources are unlikely to be impacted. As I mentioned, it emphasises the importance of patchy fires. So these burns in the Lamb Range are carried out really early in the dry season, and this means that they don't get out of hand, they don't burn extensively. And yeah, they do leave these patches of grass behind. This is hopefully going to feed into the management of that small population a bit further north at Mount Spurgeon. So that is a habitat that
Chris, time is almost up.
No worries. That hasn't been burnt as frequently. And so once managers can get fire into there, then hopefully these results feed into that. And yeah, stress the importance of low intensity early season burns. But we really don't know how they might respond to high intensity fire. And that's where that Mount Spurgeon site, which hasn't been burned as frequently, could be an important study site to monitor bettongs across what could be a high intensity fire when burning does happen there. So yeah, acknowledgements to a lot of people. It's a very collaborative project. Thanks all of you for listening, and yeah, happy to answer any questions.
For presenting my research. So I'm going to talk about the Wolbachia, one of the most invasive symbiont that we know on Earth, and also it's a unique feature of the host shifting. So Wolbachia is an alpha protobacteria widely distributed in arthropods and nematodes. It can maternally go to next generations and infect the developing oocyte. You can see here the Drosophila cells, the green one, and also the red Wolbachia around them. Scientists love Wolbachia. They study Wolbachia for a lot of good reasons. First is the master of manipulations. It can manipulate the host biology and reproductive systems in various ways to increase its own transmission chances, such as inducing cytoplasmic incompatibility, male killing, parthenogenesis, and feminizations, and even sometimes boosting the host immunities. And in that case, Wolbachia have several applications in our fight against the vector-borne diseases. The second feature of the Wolbachia is a high abundance and diversity. So more than 40% of all arthropods are estimated to be infected with the Wolbachia. And when we talk about a Wolbachia, talking about 1,000 strains that so far found and categorize it into 17 supergroups. I'm going to focus mostly on supergroup A and B, which are reproductive parasites of arthropods, and have the third feature of the Wolbachia, which is the ability to do the host shifting. So we can conceptualize the host shifting into distinct steps. First, the Wolbachia cells should physically transmit it from a donor to recipient host, and this process can be facilitated by the biological vectors. Then they should proliferate in the new host and reach the germline and do the vertical transmissions, go to the next generation and spread within the host populations. So I recently published a review papers and talk about different aspects of the host shifts. Now I'm just here want to point out one of the most controversial questions that we have in Wolbachia host shifting, not only Wolbachia, I can say the any host parasite system. And that's this question is, why a particular strains or a parasite shift from a species, let's say A to B, but not C. If you have these three species, let's say the A, B, and C, well, that you can imagine the human, chimpanzee, and a dog. And if we have the given phylogeny of these, three species, species, well, A and B, as you can see, they are mostly closely related. In this example is human and chimpanzee, and they are alike in many aspects, including their inner physiology and immune response. So we expect the host shift happen from a species A to B. We call this phylogenetic distance effect. But if we have a distribution, the geographic distribution of these three species, and consider their ecological niche and geographic overlap, in that case, if we understand that the A and C are most closely related, for example, the human dog, that's they almost have the identical geographic distributions, then there is a higher chance that they get involved into the direct and indirect physical interactions, and the host shift we expect to happen between these two species. So which of these play the most important role here, geography or the phylogeny of the host? I'm going to test it on a scale insects in the case of the Wolbachia infection. So there are sap-sucking insects with a huge variation in the biology, but also they have also very diverse relations with the ecological components in their environments, like the host parasitoid interactions that they had, and also the predators, but particularly they are in very close interaction with ants, because they can induce the honeydew, and this honeydew can be consumed by ants, and well, in return, ants provide some level of protections. This high level of trophaxis interactions can be a way to transmission of the Wolbachia. So to study Wolbachia host shifts, we had the previous study, and in that study, we understand the infection rate of the Wolbachia. Thanks to the COVID, now we that everyone can understand the difference between the Wolbachia, or let's say the infection of the determination of the infections and strain determinations. So by just the PCR test, that you can understand that you have a COVID. But if you want to know what type of COVID we have, you should do the sequencing, for example. So here is the same. We already did our PCR on all the samples of the scale 6 species we collected, particularly from Queensland. And then the next step was the sequencing to understand what type of the strains we have. And then, so after that, understand the pattern of the host shifts, and to see the effect of the geography and also the phylogeny to explain these host shift events. However, it's not that much simple, because to do the accurate strain determination, there are some difficulties in the case of Wolbachia, because more than one strains can co-infect the one host. So we come into the to develop a new methodology based on Illumina pooled amplicon sequencing. So we pooled the amplicons of the Wolbachia, which are the non-overlapping regions, and also the host genes, and all of them, so pool it per sample and send them for the myosecretes. And the results of this kind of the sequencings is a soup of all the sequence that we're looking for, and the strain determinations will into that, so require developing a bioinformatic pipeline. So the second step was develop the bioinformatic pipeline. It has a lot of details. I'm going to skip that and just tell you that the pipeline was powerful enough to, out of all this mess, accurately determine the strains that we have, especially in a case of triple infections or double infections that we had. So now we know each species that we have in our system, each skeleton species will infect them with what type of the Wolbachia strains, what Wolbachia strains we have. So we have a pattern of the host shift around. And the next question is how to understand the role of the geography of the host, and also—
Time is almost up, Ethan.
Sorry?
Time is almost up, sorry.
Yeah, sure. So the next step is a generalized adapted, so the mixer model. We just put all this information into the one model. So on the top, you can see the Wolbachia phylogeny, so all the Wolbachia that we found in the systems, and also, well, on the left, you can see the skeleton six that we found, and the squares here are actually the infections that we found in each species. So this is a pattern of the infections that we have, and also the model said that so phylogeny played the most important part to explain the Wolbachia host shifting, especially at the genus level. So, and this is also under publication well in the molecular ecology. So we found the NGS pooled amplicon sequencing is accurate way, and we developed the pipeline for that, which is easily to use by anyone. And the host phylogeny can explain Wolbachia. Well, distribution is skeleton 6, and the things that I didn't talk that much is the rule of the ant in the skeleton 6, the infection with Wolbachia and the systems, which we found some evidence of that. Thank you very much for your time and listen. If you have any questions, I'm more than happy.
So they start by extracting DNA with the normal techniques, quantifying them, and then trying to retrieve the profile. And the profile counts on PCR amplification and what we call it first generation sequencing, where they look at short tandem repeats inside the DNA to identify different humans and link them paternally and maternally to their families. However, when you talk about samples, even if it's 100 years old, we call them historical remains or degraded remains because the DNA starts to degrade once the person dies. And given that those soldiers have not been fully buried in good conditions, especially the ones in PNG, the remains start to degrade very quickly. So the quantity of the DNA that we get from the remains usually is very, very low, and the quality is really, really bad. So the DNA is highly fragmented. Very, very rarely when you get long chunk piece of DNA that you can count the short tandem repeats inside it and could get a profile. I was approached while I was still in Griffith to do a pilot study where there was a World War remain a remain from World War I that was found in Belgium. And they wanted to see if ancient DNA can bring more information to a study of the fallen soldiers, and can we identify more information that helps to repatriate those remains. So basically, usually the difference between ancient DNA techniques and the old techniques that every forensic person uses in the forensic labs is that they still count on the PCR for amplification. However, if you have a very degraded DNA that has a lot of inhibitors, a lot of stuff that prevent the DNA from being amplified and detected, you usually get a failed PCR amplification, which leads to no information at the end, or very minimal information that really can't be used for profiling those remains or identifying relatives. However, when you look at what we use for the ancient DNA techniques, we started to use the second generation of sequencing, where we build libraries. And while we're building the libraries, we introduce more correction for the damage that happened in the DNA, and we try to get the DNA to a better shape and try to increase the quantity and improve the quality of the DNA. So ancient DNA analysis allowed us, using the second generation sequencing, to do human identification using whole genome, the whole genome of the person, his maternal DNA, the mitochondrial DNA, and his paternal coming from the Y chromosome, counting on the single nucleotide polymorphism. So instead of counting on repeats, long repeats, that's not possible to achieve from the remains, we count on just one change in the DNA in different places to make a profile for the person. It also allowed us to detect, using those single nucleotide polymorphisms, to detect the phenotypic characteristics, like, for example, what eye color that person might have, and what color of skin, what color of hair. All of those information were possible with the ancient DNA techniques. And the reason why we were after those informations from the World War remains is that we have some, when they went to the war, they identified, they wrote about themselves, what eye color they have, what hair color they have. And if we can match those records to some old photos and some of those records, we can link the individual that died in the war to a person and to his family. Also, we use the ancestry SNPs to look into where they were coming from, most likely which part of Europe they were coming from, and try to identify the family, the current family members. To increase the chances to get more DNA out of the remains, couple of, like 10 years ago, we started developing technique that capture. It's basically like if you go for fishing and you want to capture a certain fish, you use the right baits. We do the same thing. We design baits for the human DNA that we're after, and we use those baits to capture the human DNA from everything else, all the contaminant DNA, all the environmental DNA that comes with the remains, to increase our chances and the amount of information that we get from those degraded remains. It also saved us a lot of money because second generation sequencing is really, really costly. It takes a lot of money to get more and more data. So when we use those target capture techniques that minimize the amount of background noise and other DNA that we don't need, and increase our chances and minimize the cost that we pay for getting the DNA. For the sample that I tested, as you see, it was wonderful, well-preserved sample. It was coming from Belgium.
Time is almost up, Sally. Thank you.
In comparison to all other remains that I worked with from Australia. So we got complete mitochondrial DNA, which was highly covered. We got Y chromosome, and both of them directed us to that individual is maybe coming originally from Scotland. And then we did the ancestry to know from which population, and it fit within the European population, but it also told us that that person had a blue eye, and he has like dark blonde hair, he had fair skin, and he had some freckles on his face, which allowed the police, the undercover war casualty unit, to compare it to the records and see if they can identify the person. Thank you. If you have any questions, I'm happy to take them.
Thank you, Sally. And
Fascinating.
Yeah, that was incredible. And thank you again for delivering the talk despite the technical difficulties.
That's all right, thank you.
Would anyone like to comment or ask a question on Sally's talk?
Great talk. Great.
Thank you.
We've got Katie. Katie would like to ask a question. Go on, Katie.
Yes, hi. I was wondering if anyone who had missing family members have submitted DNA that you could potentially match to.
So that was exactly my other thing that I wanted to say, but time didn't allow me, which is basically the drawback from that preliminary or pilot study was that I didn't have a database of family members to compare it to. And that's exactly what we're doing now at QUT in our big funded project, that we're working on building the family members database or reference database from their DNA to compare it to other remains as well. And hopefully that will give us better information. So we already started contacting the relatives, and we're hoping that we can match their DNA to the remains.
That's brilliant. It's such important work. Thank you.
Thank you.
Yeah, that's excellent. I just wondered, for me, in my head, ancient DNA, that's always sort of associated with an archaeologist or a paleoanthropologist, someone who works, with the dirty bones and things that they've been in the ground for thousands of millions of years. And so would you kind of, just broadly speaking, would you say that the understanding of what ancient DNA is and, how to obtain it, would you say the understanding of that is getting better among police forces and forensics, or is it still very much kind of, oh, just send it to the archaeologists, they'll deal with it kind of thing?
It's early days, but that pilot study was the start. So that was basically the army approaching me or the defense force approaching me. Can you trial those with those remains? Because usually with the forensic techniques we didn't get much information. Can you give us more information? So we started gradually to start offering our help with the forensic techniques. We are not replacing the forensic techniques, but we're just giving them more power with the data that we can identify. But we still work with the old bones, and for us, those old war remains are a bless because they are not that old.
Yeah, excellent. Thank you. That's really, really significant work. Definitely. Thank you.